Today, as we come to the end of our series with episode number 13, I must say I have developed a very strong attachment with each one of you through this series. CSIR Net is a marathon, and we were together in this journey while you covered 12 units with all of us. And I'm sure you are super excited to know some tips, tricks, techniques for Unit 13, episode number 13, where we are talking about methods in biology. Namaskar, welcome. My name is Shekhar Suman. I'm the CEO and Managing Director at Biotecnika. And today, we are blessed to have one of the most Buland Bharat ki Buland Beti, the most intelligent, finest expert of Biotecnika with a clarity of thoughts and voice such that we all are big fans of her. None other than Tanushree ma'am. Yes, you heard me right. Tanushree ma'am is a specialist of Unit 13, Methods in Biology. She has diverse experience across academics as well as industry. And today she's going to throw not just light on Unit 13, but she'll give you insights, insider tricks, and unique strategies, which will help you not just close in Unit 13, but definitely qualify CSIR. So allow me to welcome none other than Tanushree ma'am. Namaskar ma'am, welcome to the show. How are you?
Namaskar, sir. Thank you so much, sir, for your kind words and inviting me for your show. And especially, I'm really glad to be a part of this show, sir. So thank you for inviting me, and thank you for your kind words.
Thank you, ma'am. So, audience, that's Tanushree ma'am. And Tanushree ma'am, our audience has passed through 12 units now. And today they are watching the Unit 13. I'm sure they all have gone through a lot of concepts, and by now they all will be tired. Like, okay, Unit 13, is it really important? Should we skip? Should we study? A lot of questions they have in their mind. So let us quickly jump into Unit 13 by asking you one simple question. If an average student who has never studied Unit 13 from CSIR net point of view, how difficult this unit is on a scale of 0 to 10, 10 being the toughest and 0 being the easiest. What's your take on that?
Yes, sir. Actually, what I have observed is that at the very first glance, then when students see the syllabus, they think that unit is very difficult. But if you ask me, then I will say that it's, if not very easy, not very difficult also. So on a scale of 0 to 10, I will say that it is around 7 to 8. 7 to 8, I will like to give. Because usually if the questions asked in part B, they are very direct and they are memory based. So one can easily score, they can easily score marks in part B. However, yes, part C questions, they require, like they are experimental based questions. So they require conceptual clarity, keen observation and analytical skills. So yes, they are a little bit difficult, but if the students, they follow proper strategy, if they prepare this unit with proper planning, definitely they can score very good marks in this particular unit.
All right, so there you have it, guys. It's tough, no doubt. Not very tough, but yeah. But the next question will be, okay, it is tough, but should I get into this battle or not? Should I study this or not? Can I skip it? So the next question to you, ma'am, is, okay, 0 to 10. 10 being topmost priority, 0 being not at all a priority. Tell me how important this unit is for CSIR.
Sir, I will say 10. It should
10.
be on priority.
Oh,
yes,
that's
sir,
a 10.
It should be on priority. Yes,
Highest
100%. I must say 200%. I can say one should never skip this unit. They have to prepare this unit. I'll give you the reasons. I have basically identified 3 reasons that one should not skip this unit. The very first reason being, sir, that it is very much important from exam point of view. Lot of questions are asked from this unit, and the questions asked are not only direct, but even questions are framed by combining topics from other units as well, like unit 13 and unit 1, or unit 2, unit 4, unit 8 even, and unit 12. So if, like, for example, if someone is preparing unit 2 cell division, and in the exam the question is framed by combining topic cell division and flow cytometry technique from unit 13. So if you have prepared cell division, it will be of no use. right? They need
Right.
a knowledge about flow
Flow
cytometry also.
cytometry also.
Yeah, and questions are very scoring. So if they know
Oh,
yeah, if they know about the technique as well, they can easily answer the question. Then
wonderful,
second reason
wonderful.
is, sir, yeah, the second reason, sir, that I feel is most important is that the students should see at a broad level, right? They have to
Hmm.
even think about their future as well.
Right.
After qualifying the CSR net exam, when they actually enter in the research field,
Mm-hmm.
these are the techniques, these are the instruments they have to actually deal with.
Correct.
And
Absolutely.
specifically, yes, sir, and specifically when they're actually entering into research field, even if they have to make the research proposal, even if sometimes when they are applying for research positions in the interviews, sir, the questions are asked on the
Acha.
techniques. So that
Right.
is why I will say from practical aspect, this unit is again very much important. And third
Mm-hmm.
reason is, sir, that if the student is preparing for other exams as well, like GATE, DBT, then most of the topics of this unit 13 from CSIR net are common with other exams as well.
Other exams as well. Right.
Yes, sir. DBT, ICMR,
Right.
GATE, all.
ठीक है, ठीक है. So there you have it, guys. Ma'am answered you and gave you a 360-degree view of why you should study this unit. I didn't ask, but you see, her insights are very valuable. And that's why we have a special expert today. All right. So now that we know how important this is, ma'am, next question. What should be the approach to study this unit? What do you think?
Sir, you have asked a really very interesting question, and believe me, this is what, like, students usually they don't know how to approach the questions or to approach this unit actually, and that is
Right.
why they even this unit is scoring, they are not able to score well in this unit. So,
Correct.
if talking about the questions specifically, one should always always read the questions carefully. That I always say, read the
Mm-hmm.
questions carefully, see what are the information, what is being asked in the question. Is it direct, is it correct statement or incorrect statement is asked, or any ascending or descending order arrangement is required, or in the question, like if it is a graph based question, then they have to understand that they don't have to think out of the box, right? They
Correct.
have to see what exactly the information is given in the question. Based on that only they have to answer the question. So first point is read the question carefully, think on what concept that question is based, and then
Mm-hmm.
once they recall the concept, like either they can, like if it's a uh, numerical based question, so recall the formula, just see if any unit conversion is required, right? And third point is once you recall the pathway, once you recall the working, let's say if a question is on working of the technique or principle or the formula, then try to apply that particular concept. And sir, definitely to to score good marks in this unit, one has to practice a lot of questions. I will say the strategy for studying this unit should be that you should see the question paper first, you should go through past year question paper first, see what type of questions are asked, and then prepare the topic. So that will be
Wow.
very much helpful.
You actually answered my next question. I was about to ask you that should we study theory first or look at the questions first. So you answered that question. Brilliant, wonderful. So, ma'am, now that we know, okay, the approach to solve, to study this particular unit, the next question to you is, okay, out of all the units and all the experts I have interacted, you have given a rating of 10 out of 10 on a level of priority for this unit. And that means it's very important. And if this is very important, should we not start studying when we are actually starting our preparation for CSIR? And should we not start, say, unit 1 and 13 together or something like that, so that it is easier to, because by the time we have finished 12 units and reach 13, we'll be definitely tired and that sequence will not work. So don't you think we should actually start studying unit 13 very early in our preparation?
Yes, sir. I would like to. I'm really glad to answer this question. Actually, I will say that if the student directly start preparing for Unit 13, the difficulty that student faces, they don't understand Unit 13 at a first glance, is because their basic foundations are not clear. So I will not suggest that one should start Unit 13 on a very first, like first unit should not be Unit 13.
Okay,
First, they should
fair enough.
Yes,
Build
we
the
should first
basics.
emphasize on first four units, sir. Unit 1, 2, 3, and
Two,
4.
three, four. Okay.
I'll give you a
Okay.
reason, sir. Like for example, if a student is preparing for immunological technique, right? But if the
Mm-hmm.
student don't know what is antigen, what is antibody, and yes, it happens. Students
Difficult.
actually they don't know such basic concepts as well. And I will not blame students for that. It's our education
It's our
system that in
education
the
system. Yeah. Yeah.
yeah, actually
I can go
in
on
the
about that.
graduation.
Right. Right.
So the point is their basic foundation
Mm-hmm.
should be there. So I will say like unit 1 is all about basics. So unit 1
Right.
they should study, then 2 they should be familiar with the structures, cell division process, the structure and function of organelles, then third and fourth unit, and then move on to unit
Then,
13. So first four
okay.
units if they are done, then fine, they can start with unit 13. But
Okay. Okay.
at the
Fair
very first
enough.
step I will not say. Yes, sir.
All right. All right.
Very
So,
first
ma'am,
unit
now.
selection should not be unit 13.
Okay, all right, perfect. So it's an advanced unit, so we should not start jumping directly here. First, finish the basics, then move in here. All right. So now that we know this part, what are the standard books we should refer for this particular unit?
Yes, sir. Standard books definitely students should refer for Unit 13. First of all, I would like to say this point because what I have observed, what I have analyzed on going through previous year question papers, that questions are directly like if a diagram is given in a book, they directly pick the diagram and frame the question from the standard books. They pick the diagram, they pick the examples, and they frame the questions
Directly.
from unit 13, right? So I will say that there's no one single standard book they should refer. I will suggest they like for every subunit there are standard books that they should refer. One standard book is fine, but yes, standard books they should refer. Especially they should emphasize on diagrams and examples which are given in the books. So first book I would like to tell you is like for preparing recombinant DNA technology, that is subunit 13A. One should can refer gene cloning and DNA analysis by T.A. Brown.
T.
For
Brown. Okay.
13A, for immunotechniques 13B, one should refer Cube immunology.
Okay.
And so no entire book is not required. Whatever the topic is given in the syllabus,
Topics.
they should refer from that book.
Selective study. Okay.
Selective study. And then 13C, that is biophysical methods, and for radio labeling techniques, that is 13E, one should refer principles and techniques of biochemistry and molecular biology by Wilson and Walker.
Wilson and Walker. Okay.
Yeah, sir. And for
Okay.
statistical methods, fundamentals of biostatistics by Rastogi, and
Okay.
for microscopy, that is 13F, one can refer Prescott, microbiology
Prescott.
Prescott. Yes, sir.
Mm-hmm.
And for GNS, that is electrophysiological methods and methods in field biology, I will say that usually, I say that they should refer PPTs only, Biotecnika PPT, and the research articles usually links I share with them.
Okay. Wonderful. All right. Okay. So now, my dear audience, you can see that like other units, maximum the experts will suggest you two, reference books. Here, ma'am is suggesting like 4 or 5 reference book. On the top of it, the PowerPoint short notes which Biotecnika provides you, along with the research papers which Biotecnika provides you. So this is like huge, right? And it's on a priority basis important, and it's not just like it became important like last 5 years. Since the time we are providing coaching for CSIR like 15-16 years now, we have seen how important this unit has played a role in preparation. So now, ma'am, we know which book to refer to. My question to you is, when we are preparing notes, what should be our approach? How should we actually go in preparing notes so that it is easy for revise, recall, and other stuff? What's your take on that?
Sir, I will say that mind maps are the best for preparing Unit 13. Mind maps
Right.
are the best. And like mind maps, like they should be like, even if the student is making notes, they should be like, sir, the point is
In a
the
flow
students, they
chart or a
don't
mind
know.
map.
Yeah, they basically they don't know how to make notes. So I will
Right.
just say notes doesn't mean rewriting the books. They should
Exactly.
understand that either whatever every student has their own approach. So if they are making flow chart, it should be precise, right? But because in a technique you have to study lot many things. You have to study basic principle also. You have to study basic overview of the process also. If any formula is there, that also you have to study, and application. So mind maps are the best. And try to refer more and more diagrams, like pictorial diagrams they should refer to understand a particular technique, right? And animated video, sir, I will say that if a student is preparing, they should definitely refer animated videos to understand the working. Because actually what happens, sir, that most of the students, they don't know how UV visible spectroscopy looks like. They have never seen flow
Right.
cytometry. So how they will understand what the technique is all about unless and until you work on it, you see that
Yeah.
instrument. So I will say that there's various company website, BioRed and Syngene. Many companies' websites are there. Just visit them. They have the animated video for PCR, for flow cytometry, and all. They can refer that. So
Mm-hmm.
uh, that is most important part, to relate what you are studying. You should relate. So
Right.
the
Right.
strategy is smart tools they should use, and among the smart tools, especially mind maps are the best. They can make
Mm-hmm.
formula notebook, note down the formula separately, like probability of restriction site or Beer-Lambert's law or chi-square test formula, half-life formula, radioactive decay formula. They should write separately in a notebook, and so that they can revise it frequently.
Okay.
So that,
Okay.
and compare and learn. One more point:
Yeah.
compare and learn, sir. Comparative tables they can refer.
All right, all right. So now, ma'am, we know how to make the notes, and you suggested the smart tools. I'm sure you're talking about the Conceptica notes, Conceptica
Yes, sir.
flowchart, and all
Yes, sir.
that stuff, and mind map, which Biotecnika recently released.
Yes, yes, sir.
Audience, if you want to download a free sample of those, the link is given in the description or in the comment section. Please check that out. So now, ma'am, we know the notes part, right? And one more thing I want to, remind the audience, all the people out there, that your notes should not be like all our experts have said, and this is my conclusion also, that notes should not be an expanded form of your book. It should be crisp, concise, and a consolidated form of the book so that it is easier for revision. And that brings me to the next question, ma'am. When students are revising, what should be the strategy according to you?
Sir, I will say that for revision also, like when
Mm-hmm.
they are revising, the first thing is go through question paper. Go
Right?
through question paper, see what question is there,
ठीक है.
and try to recall what concept is applied there.
Mm-hmm.
And
Mm-hmm.
then, based on that concept, revise that particular topic. Like it's not
Okay.
that revise the topic and go through the question. I will say go through the question first, and then
अच्छा.
based
Then
the
revise.
concept, yes, then revise. So this way
Okay.
they are able to, because when they see a question, they start processing. Okay, what concept the question is based on. So they are recalling only.
Very
So
true.
that
Very
is
true.
the revision strategy. And second, they should refer smart tools, Conceptica
Mm-hmm.
Lite or Concept Notes, Concept Table, Concept Wheel, and Mind Maps, as I told you. Concept
Mm-hmm.
Notes also. So whichever they are comfortable with, they should refer that. And
Okay.
even for like if, practice as many questions as they can.
Okay.
Revision, because at the end, whatever they are studying, they have to apply that concept in solving questions only. So practice, practice, and practice. More they practice,
Mm-hmm.
more they learn.
Right, right. So, all right, guys, now you have it, how to revise. And very important thing, which ma'am has also highlighted, and I would like to highlight here. The smarter you study this unit, the better your recall value will be. And remember, it is your only tool which is going into exam. That's your brain. So your recall value should be high for any of these units, whether it is facts or logical questions. You will be able to solve them faster if you are able to recall the concepts faster. And for that, Biotecnika has developed a wide range of tools called as concept revision tools. The link is given in the description. You can check that out. That will definitely help you for Unit 13. And experts like Tanushree ma'am are always available at Biotecnika to help you, guide you, mentor you for this particular unit or any unit for that matter. All right. So, ma'am, now we'll have our next question, and that is: we now know how to make the notes. We now know how to revise, but when we are studying this unit, at the same time, we also, as students, tend to target GATE, ICMR, DBT, ARS, all other exams, right? So generally, we also advise sometimes students don't put all the eggs in the same basket, try other exams also. So multi-exam strategy, if we have to look at, what will be your take? What will be your point of view? How exactly we should study? Like, how much will be the overlap of syllabus of other exams for this particular unit?
So the very interesting part is that majority, like I will say 99% of the topics which are given in Unit 13 of CSR Net, they are exactly the same given in DBT also.
Replica.
So it's
Okay.
a replica. Exactly they are given. It's just like one or two topics which are not there, but majority are there
Majority
in
is there.
DBT. Yes. And even,
Right.
sir, in GATE, if we see section 7, which
Hmm.
involves recombinant technology,
You mean to say
subsection
Gate BT or Excel?
Gate BT, sir.
Gate BT.
Yes,
Okay. Okay.
yeah.
Please go on.
In Gate BT, the section 7, which is there, the
Right.
recombinant DNA technology subsection is there, molecular tools, analytical tools. They
Correct.
also cover the topics which are exactly given in Unit 13 of CSR Net.
CSR. Right.
Only one
Right.
or two topics are different, otherwise entire topics are like common for gate bt, for dbt, and even, sir, for icmr, because icmr
Mhm.
also, like when we talk about the techniques, they are used in medical purpose, but the only
Right.
difference in icmr, the questions which are asked, they are more like related to
Animal
the
physiology.
medical field,
Yeah. Medical field. Yeah.
medical field, yeah. So
Right.
techniques
Right.
will be common for all these exams.
Common.
So it will an
Right.
advantage for the students. Like I will say that if they're preparing for CSN Net, simultaneously if they're preparing for others, then this unit will be an advantage for them.
Advantage for them.
It
Right.
will be
Right.
common, so they don't have to devote extra time for this particular
Right.
topics of
Right.
unit.
Right. And how do you see the future of students who are going to pursue their PhD? Uh, this unit at the beginning of the show also you said this, but I want to repeat this because I feel this is an important question. When they're pursuing their PhD, definitely all these techniques will come handy, right?
Definitely, sir. Like even from my research experience, I would like to say when I was
Yeah.
pursuing PhD, if you already have some idea about a technique, right? Like I will say practical and theory goes hand by hand. You
Yes.
cannot perform practical if you don't know theory, what that
Theory.
technique is all about. And if you
Correct.
don't know theory, how will you use that technique, right? So
True.
usually students say that practical aspect, but first you should understand theoretical aspect, then only you will understand the practical. Because, sir, actually when you perform the experiments using the techniques, it's different. There are a lot of troubleshooting you have to do, you have to optimize the protocol also, right? But definitely this particular preparation for this particular unit is very much helpful, because when you are entering into the research field, nobody will tell you what is uv physical spectrophotometer. They will just
Correct.
give you that, okay, this is the instrument and now work on it. Even if you are making a research proposal, you should know which techniques are recent, what are the recent advancement in the techniques. Like in flow cytometry, we have FACS, fluorescence activated cell sorting, so we can perform sorting of the cells also. So if you are aware, if an advancement only you can go if you know the basics. So definitely it is
Right.
very much helpful, sir. And I would like to suggest one thing, sir, to my students, to all the students, that don't waste this time. Even if you're doing graduation or post graduation, you can join some workshops or certification courses, or they can attend the webinars based on these techniques, because definitely it will add on to their CV only. And especially in the
Right.
interview when they're going, this will keep you from the group, you will stand out, right? So really
Right.
it will helpful for them. Don't like they don't. And even in the colleges, if they are doing graduation, post graduation, they should take their practical class seriously. Don't bunk your
Right.
practical classes. Just see what, because microscopy, spectroscopy are common in every lab in
Right.
institutes or universities. So at least start from these techniques.
Right. True. 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 So,
Try to
ma'am,
work
now
on
we
them.
right now we know almost 360-degree view of Unit 13, and we now know that how to study, which exams we can get benefited if we are studying, and how what in the larger picture of PhD, how handy this particular unit, the theory part, will come. But let's look at this. Students study 13 units, or say 10 out of 13 or 9 out of 13 to write this exam. And definitely they must study this. They all feel a lot of pressure and tension, a lot of anxiety. And now that the exam is very near, they all want to, know some, tips, techniques, and tricks from you. How should they ease up the anxiety? Because I have seen you are an expert of guiding students and mentoring them. So I'm asking you this special question, that how should they reduce the anxiety when they're dealing with all these difficult units?
Yes, sir. This is a very important question, and especially I can understand. We on our daily basis, we come across a lot of queries from our dear students, right, regarding this special preparation before the exam. So, very first thing I would like to say, sir, that they should be confident in whatever they have studied so far. I know
Right.
it sounds difficult, but trust me, like they have to be confident. Just they have to sit peacefully and they have to tell themselves that yes, I can, I will
Mm-hmm.
uh, score good marks in CSR net exam, I will qualify CSR net exam. So they should
Right.
have faith in themselves, they should believe in themselves. So I will say to
Right.
the students that believe in yourself. When you
Mm-hmm.
believe, automatically you'll attract the positive energy towards you, and definitely you all are so knowledgeable. Just you have to be focused at that time of exam. So I will say, just at this particular time, just before the exam, just take care of your health, eat healthy, keep yourself hydrated, revise.
Mm-hmm.
Revision is very much important. Whatever you have
Important.
studied, revise. Don't start anything new, because you
New.
won't be able to complete. Like mostly
It will
students think that if
confuse
it,
further.
yeah, they will confuse. They think that if I'll start revision like this particular topic at the end, I will be able to remember, but they end up getting confused.
Doesn't happen.
So
Correct.
yes, sir. So don't start anything new, and just believe in yourself, and just take care of your health and revise whatever, and practice question. Practice
All
as
right.
much questions as you can.
All right. So there you have it, guys. Thank you so much, Tanushri ma'am, for joining us today. There you have it. You have now got a 360-degree view of the entire Unit 13. And through this series, we have covered the entire syllabus of CSIRNet through Biotecnika's What to and How to series. One very important point ma'am made in the last question, that is believe in yourself. I would like to add one important line to this. If you want to boost your belief in yourself, then take guidance of true mentors, true gurus like Tanushri ma'am and all other teachers at Biotecnika. Remember, science is an interesting subject, but if you're not enjoying it, then probably you are reading from the wrong teacher. Come fall in love with science, come fall in love with biosciences with Biotecnika. Today, we end this show with Unit 13, 13th episode. I'll see you soon in our next series and next video. Till then, please take care, study well, make it large, and do let us know once you make it large. It was wonderful interacting with all of you through this show. And as I bid adieu to all of you, let me tell you, we have a special relationship. Let's continue by subscribing to Biotecnika and be in touch. Any questions, put them down. We will definitely answer. Tanushri ma'am will specially personally answer to all of you. Thank you so much. Take care. Bye-bye. Dhanyavad.